we are talking other day that, cyanobacteria-like organisms became a chloroplast in the process of endosymbiosis, right? And for billions of years, became a chloroplast. So, as I told you, once a living organism like cyanobacteria, which becomes endosymbiont, lives in a host cytoplasm, it doesn't need many genes anymore, right? Because it is living on a host cytoplasm. So he provided all facilities, right? So therefore it lost many genes completely. So you can compare cyanobacterial DNA and chloroplast DNA, much smaller than the cyanobacterial DNA. Okay, it's very, very small in size. We lost many genes. Okay, but then also not only completely lost, nucleus also wanted to have a central command over the chloroplast. Okay, and our same goes to mitochondria too. Okay, so then what happened? Then many genes were transferred from the nucleus, from the ancestor genome, that is cyanobacterial genome, to the nuclear genome. Okay, so the protein that are coded by the chloroplasts before, by the cyanobacteria before, is now being coded by the nucleus. Okay, and once it's coded by the nucleus, the messenger RNA is produced in the nucleus, then it comes out to through nuclear pores to the cytoplasm, where it translated on ATS ribosomes, and then post-translation are imported into the chloroplast. Okay. And still there are some proteins that are coded by the chloroplast genome also. We are talking about its other day, right? RBCL is the small subunit is coded by the nuclear genome, and large subunit coded by the chloroplast genome, okay, Rubisco. Therefore, Rubisco has been Rubisco small subunit because major protein being transported, because 50% lipid, I told you, and 8 small units and 8 large subunits form one enzyme called Rubisco. Okay, so to build one enzyme Rubisco, with 50% protein, chloroplast protein. Okay, Rubisco is the most abundant protein that is transported from the cytoplasm to the chloroplast. Okay, therefore most of the model studies, they look at it, look at Rubisco as a standard. Okay, from standard protein. Okay, for its translocation mechanism. Okay, all right, understood. There are many other genes we are not talking. I will talk about some, but I may not talk about it today also, depending on time. Okay, I don't have only 1 hour today. So there are also many other genes which are not only transported to the cytoplasm, they are also transported to the thylakoid membrane, like light harvesting protein complexes, LFCs, right? They are transported to the thylakoid membranes, and some are transported to thylakoid lumen, right? Thylakoid lumen, okay? So oxygen evolving complex proteins, right? 33, 23, 18 kD proteins. They are all coded by nuclear genome. And post-translational have imported into the lumen of the thylakoid. So it has to cross the outer membrane, intermembrane space, inner membrane, then reach stroma, then it cross the thylakoid membrane, then it is the lumen of the thylakoid. Okay? But think out, there are many barriers for it to reach into the lumen of the thylakoid. So therefore nature has evolved different kinds of mechanism, okay, to import different kinds of protein, okay, into the chloroplast. But the topic that they given to me is membrane, right?
Sir, I have a doubt.
Yes.
You told that the genes have been transported from the chloroplast to the nucleus, and then
no protein grows out from the chloroplast to the nucleus.
You said from the time of cyanobacteria, some of the genes are being introduced by the nucleus now.
Yes.
The chloroplast. So what is the advantage of it? Because importing it back to the chloroplast is expensive first.
The advantages is this, that it has Mendelian inherited now, Mendelian fashion. If you remain in chloroplast, it will maternally inherited. Okay, that is one of the reasons why nature selected to put all the genes back into the wherever possible, back into nuclear genome for Mendelian inheritance. Okay, that is very important. Mendelian inheritance is very important component of plant breeding. Okay, cross breeding, cross pollination, right? We have come out. If you look at it, when in the process, nature has favored cross pollination. Okay, went out of the self pollination to cross pollination. Cross pollination happened among two different same plant, two different plants later on, right? And not only that, then plant became dioecious. Male plant, you see papaya, there are male papaya and female papaya plants. You know it? Male palms and female palms. You know it? So it has moved slowly from hermaphrodite condition, right, to the dioecious conditions. Okay, all right. So same thing also in animal evolution. Okay, we started with annelids, right, which is where mostly self-fertilization, annelids, right. If you come to that, still there are strains of but very lower organisms, we have self-fertilization in animal system. We have come out of that, and always there is cross-fertilization. Okay, you have men and women. Okay, there are no. Okay, so that has happened. The nature has favored this cross-fertilization to segregate gene pools, right, and then remix gene pools. Okay, so that evolution can go on. Okay, so this cross-pollination has favored evolution. Okay, all right. Okay, so when let's say, there are two kinds of protein import. One is co-translational protein import, another is post-translational protein import. What is co-translational? As and when protein synthesized on the ribosome surface, okay, right? The proteins, it comes a signal sequence. They recognize endoplasmic reticulum, docks to endoplasmic reticulum directly, while it is still being synthesized in the ATS ribosomes. If you see most of the protein synthesized, all the messenger RNA or polyribosomes are bound to the endoplasmic reticulum. So as and when it's synthesized, the signal sequence of the protein docks to the endoplasmic reticulum or binds to the endoplasmic reticulum, then goes into the endoplasmic reticulum for post-translational processing, methylation, whatever takes place, the protein post-translational modification, alkylations, etc., all takes place in the endoplasmic reticulum. Then it comes out of endoplasmic reticulum, okay, whenever required, and it depends upon destination, okay. So remember, there is no problem, okay, because synthesized and transported. You see, protein has to, if it's synthesized in the cytoplasm completely, before it attaches endoplasmic reticulum, okay, they must attain its three-dimensional configuration, right? It is a cytoplasmic aquatic environment, right? Not hydrophobic environment, but aquatic environment. So protein folding will take place, no matter what, okay, in response to the aquatic environment of the cytoplasm, okay. So protein folding has to take place. And then protein has to partially unfold to go into the chloroplast. So there are many things we'll talk about it. So I'm not talking about co-transference protein import, mechanism and all, okay? That a completely different mechanism for the transport of proteins in the endoplasmic reticulum, okay? Okay, if you see, as I told you, polyribosomes are attached to the where? Endoplasmic reticulum, okay? But if you see chloroplast or mitochondria, no polyribosomes attached, polyribosomes are not attached to neither chloroplast nor mitochondria, okay? All right. So, protein synthesis is taking place in the cytoplasm first, and then they are being imported into the chloroplast or mitochondria, okay? So, both of them have therefore post-translational protein import, okay? All right, understandable? Okay, this is, I always, so this first time it's an endosymbiotic origin, right? Chloroplast DNA is a plastome, okay, contains 300 proteins, 100 proteins encoded by chloroplast DNA. Out of 3,000 proteins, 100 proteins encoded by chloroplast DNA. So, minuscule amount, right? So, 2,900 proteins are coded by the nuclear genome and post-translation reported into the chloroplast. Got it? Okay. I'll skip the slides. All you know about light regulation of chloroplast. I think Dr. Takahashi will talk about light regulation of chloroplast and all. So I'm not touching this point. Okay. Okay. So this is the protein input apparatus present in the chloroplast membrane. You see, we are simply talking about protein that is going out from the cytoplasm here to the stroma here. This is thylakoid membrane. Outer membrane, bilayer outer membrane, bilayer inner membrane, okay, and then the stroma, okay. So the protein has to synthesize in cytoplasm, post-translationally has to be imported into through the outer membrane, through the intermembrane space, to the inner membrane, again to the cytoplasm, stroma, okay. So what it has? If you see protein, all the proteins are synthesized with the transit peptide, okay. You see, red color is transit peptide. Just like signal sequence for the endoplasmic reticulum, here you have called transit peptide, okay. This transit peptide recognizes where it has to go, okay, and receptors present in the thylakoid membrane, okay, or in the envelope membrane. Here is envelope membrane, okay, envelope membrane chloroplast decides which protein to accept, okay. So mutual selection of the transit peptide and the chloroplast. You see, both chloroplast protein and mitochondrial protein are synthesized in the cytoplasm, but only chloroplastic protein goes to chloroplast and mitochondrial protein goes to mitochondria. Cytochrome C does not go to chloroplast, nor Rubisco small subunit goes to mitochondria. Not it. So there is selection process. Why is it so? We'll talk about it later. Okay. All right. So this is a transit peptide, and this is a protein. Okay, this protein is attached to HSP90 and HSP70. What is HSP? Heat suck proteins. They will work like molecular chaperones. I talked about chaperones a few minutes ago, last class. This is the purpose I talk about is this, chaperones, okay. We need chaperones for providing proper conformation to the protein, okay. So here, these two chaperones, okay, all right. Can you see here? If you cannot come to here, come here. Come here. Why you are sitting backwards? Those who are sitting back, come out. Here, come here. There is enough space. Come here. Yeah. Okay. You can, if you want, you can come also. Okay. So HSP 70 and HSP 90. These are the molecular chaperones. Okay. And these molecular chaperones keep the protein in transport competent form. Okay. If the protein completely folds and folds anomalously, okay, without molecular chaperones, it becomes transport incompetent. If after it's synthesized, okay, in ATS ribosomes, okay, attains 3-dimensional configuration without molecular chaperones, then it is transport incompetent. It cannot be transported into the chloroplast. And therefore, these are degraded. Any protein that does not bind to the appropriate molecular chaperones, they are transport incompetent and they are ultimately degraded by proteasome system. Okay? They are ubiquitinated and goes to, and then they go to proteasome complex where they are degraded to individual amino acids. Okay? All right. We'll talk about it later on. So now you can see this import apparatus. What is their import apparatus? It is the term TUC. TUC stands for translocon in the outer chloroplast envelope membrane. TUC. Translocon in the outer chloroplast outer envelope membrane. TUC. TIC stands for translocon at the inner chloroplast envelope membrane. Okay? So TOC and TIC. TOC stands for outer membrane, TIC stands for inner membrane. So if you say TIC 20, you immediately understand it present in the inner membrane of chloroplast. It's a TOC 75 or TOC 34 or TOC 159. They are present in the outer envelope membrane of the chloroplast. Okay? All right. Same goes to mitochondria. Instead of mitochondria, you get TOM and TIM. Okay? TOM is the chloroplast outer membrane of mitochondria. TIM is the inner membrane of mitochondria. Okay? Okay. So, TOC 159, TOC 34. Has anybody taught you about anything about this import apparatus in this class, in this lecture series? Huh? Okay. All right. Okay, doesn't matter. Okay. So, then what happens? Well, let me describe the process before you go. Okay, details. So, what happens? First, the transit peptide comes and binds to the TOC 159. TOC 159 through TOC 34. So both TOC 34 and TOC 159 work as receptors, okay, to recognize the transit peptide. Okay. Only if it is chloroplastic transit peptide, okay, they will bind with it. If it is mitochondrial transit peptide, they will not bind to it. Okay. So these receptors have the ability to discriminate chloroplast transit peptide and mitochondrial transit peptide. Understandable? Okay? All right. So once it TOC 159, then it goes inside. We'll talk not I'm describing here now. The TOC 75, which is barrel kind of protein, TOC 75. So it has a pore. When you pour inside, okay, barrel, forms a barrel in the membrane, okay, because secondary structure of the protein, hydrophobic core of the protein, so it goes into membrane and makes a barrel structure. So TOC75 can go in, so that transit peptide can go in through the TOC complex. Okay? Once it comes out TOC, then it has to enter the intermembrane space and then come TIC complex, TIC20. Okay. So think about it, two membranes, two independent membranes, outer membrane and inner membrane. Look at me, okay? Not a slide. The outer membrane and inner membrane. TOC complex is here. No guarantee that TIC will be here. Same place, same point. Because they are all moving in the membrane plane, dynamic in nature. TOC is moving and TIC is also moving. Okay? Okay. So once it passes to TOC, TIC has to align with it. Then only it can go through. Okay? So this alignment is done by TIC22 and a protein called POTRA. POTRA is extension of that, but mostly TIC22. TIC22 binds to POTRA, who is extension of TOC75. Okay? All right? All right? And TIC22 are present in the where intermembrane space. Do you see that? TIC22 present in intermembrane space. So this POTRA is hanging. Okay? Whenever sees TIC22, okay, bind to TIC22, okay. So transit peptide has come, moving in the along membrane plane, okay, like this. Then POTRA is just extending to the intermembrane space, and then to wherever you find TIC22, it binds. Once TIC22 binds, then it aligns with the TIC20 intermembrane space. Okay? So TIC22 is a very important component present in the intermembrane space, essential for protein transport. Okay? Now this TIC22, once these TOC75 and TIC20 align, okay, then you can see that it passes through the TIC20 complex. Okay? And then what happens? As soon as it comes out, it binds with CpHSP70. This is HSP70 here, CpHSP70. Chloroplastic, Cp stands for chloroplast. So chloroplast also, stroma, has also HSP70. We call it chloroplastic HSP70. Okay? All right. Binds to it is also molecular chaperone. Okay? Binds to this. Then HSP70 binds to HSP90C. There is HSP90. There are HSP90 are many kinds of HSP90. So C, the 90A, 90B, 90C, and you bind to HSP90C. Okay? All right? Once you bind to HSP90C, then it binds to HSP93 and CLPC. What are these do? They are motors. Their function here, as soon as it comes out of TIC20, okay, and there are these, you see these anchored to the membrane, right? These guys, TIC110 and TIC40. So they're simply as anchoring it, anchors, staying there, okay? And these guys are attached to this anchors, okay? What are molecular motors. They start moving, okay? What do? They pull the transit peptide. Once it comes out, as soon as transit peptide docks, binds to CLP-HSP70, okay? As soon as it binds, these two guys, HSP90C and 93 CLPC, moves like a motor. Okay? When it moves like a motor, if you see, it will pull it, right? You move with it, you bind to it, and when it moves like this whole protein will be pulled back. Okay? So then this protein comes out. As soon as it comes out, the transipeptide is clipped by the stromal processing protein called SPP. Stromal processing protein. It recognizes where transit peptide ends and mature protein begins. Okay? All right? So transit peptide is clipped and then destroyed. Okay? You can then destroy it. Okay? And the protein comes out and folds into a mature protein. Okay? So what is it? Transit peptide. What is transit peptide? A protein that then, usually, a protein that must cross the envelope membrane, both the envelope membrane, reach the stroma, should have a transit peptide. And so transit peptide, for Rubisco, transit peptide is 20 kD. So total protein is 20 kD. 14 kD, as I told you, Rubisco small subunit is 14 kD. Okay? But synthesized at 20 kD precursor. Synthesized as a 20 kD precursor protein. Okay? So if you talk about precursor protein, so this is a pre-protein or PRSS, precursor of small subunit. PRSS. As an example I am telling. Okay? PRSS. So it has 6 kD transit peptide followed by 14 kD mature protein. Okay? You understand that? The 6 kD transit peptide is destroyed after it reaches the stroma, okay, as I showed you. And what remains is the 14 kD mature protein. So pre-protein and mature protein. So 20 kD is the pre-protein and 14 kD is the mature protein. Okay. After this reaches the stroma, okay, then LSU and SSU assemble. This is SSU, right? Small subunit. Okay. LSU is already present in the stroma, coded by the chloroplast genome. Okay. So on 7 days ribosomes. Okay. So therefore, then the protein assemble through molecular surveillance and all that. Okay. This is nutshell the overall process of protein input through the chloroplast envelope membrane. Okay. This is a very oversimplification of the story. Okay. Okay, T236 super complex, we are not talking about it. T2, all together we call T236. Okay, but we are talking about that. It's more confusing for you. Okay, therefore I am saying simplification of component. Will come, I will show you later on. Okay, complexity of it. Okay, I am just only showing the very simplified process. Okay. So when you talk about this, so number one, the protein, so transit one, the pre-protein or PRSS, precursor small subunit, must bind to the chloroplast membrane, right? Without binding, no import, right? If the protein doesn't bind to the chloroplast membrane, there is no import. And where it must bind? And what is responsible for protein binding? How you know that? Whether it binds to lipids or proteins. So how it study this? What you have to do? First clone this gene of Rubisco small subunit along with the transit peptide, its own protein, all right? The whole genome, right? Rubisco gene, DNA, small subunit DNA. You have to clone it, right? Then next step is to do an expression vector. So you synthesize messenger RNA in vitro transcription, right? Then you take the messenger RNA, right? Centrifuge messenger RNA, then put into in vitro translation system. Okay. What do you do in vitro translation? You take this messenger RNA, put in a test tube. To this you add what? Rabid reticulocytes, or you can have wheat germ system. Okay. That will provide what? Ribosomes, all protein translation factors, and everything. Okay. All right. Ribosomes, right? Where the protein is synthesized, right? So you have to take, once the protein is synthesized in vitro translation, when you are synthesizing protein, you want to monitor the protein transport, right? Then you synthesize, let's say, with S35 methionine. S35 methionine. So you add, while doing translation, you add all amino acids cold except methionine, which is hot, which is S35 radioactive. Okay. So then your protein, wherever you have start codon, so it will go anyway. But wherever in between, where you have ATG, AUG, okay, it will be incorporated there. Where? Methionine. You know it? And let's say you have 6 methionines. Where? In Rubisco, small subunits. So wherever there is methionine, we have 6 radio-labeled methionine in the Rubisco small subunit now. LSU, including transipeptide if it is there. Okay. It will be there anyway, because ATG starts with start codon, ATG will be there, AUG will be there. Okay. All right. So you have now the radio-labeled protein. So now you have radio-labeled protein in your hand, right? With Rubisco small subunit along with transit peptide, which is radio-labeled now, right? Then you isolate intact chloroplast. You take, isolate the chloroplast, you might have already done my practical, you can pass the Parker gradient, okay, to isolate intact chloroplast. Once you isolate intact chloroplast, add this radio-labeled what? Transit peptide to the chloroplast. Okay, take a test tube, take some chloroplast, add the radio-labeled transit, radio-labeled Rubisco along with transit peptide. Okay, all right. So now, so priority for protein to import into chloroplast is that it must bind to the envelope membrane. You know it? So first actually binding. Okay, all right, understandable? So if it only binds to the chloroplast membrane, we just want to know how the chloroplast is simply binding to the envelope membrane or not. What you do, you reduce the ATP content. You simply go give, you need 1 millimolar ATP at least for protein transport. If you give 0.1 millimolar ATP, there will be only binding and no import. Okay, why need ATP? This ATP hydrolysis, you see. To run this motor, you need ATP, okay? If there is no ATP in the system, if you do not provide any ATP, this motor will not work, okay? Therefore, only there is binding, but no transport into the stroma. Do you understand that? Understandable? Okay. So, you give a 0.1 millimolar ATP, 0.1 millimolar ATP, very, very low ATP you give. So that binding takes place, or you can add GTP also instead of ATP. Binding takes place, okay, but no import, okay. And you then what you do? You did this assay, binding assay. This is called binding assay, right? Then what you do? You pellet your chloroplasts, okay, right? Again through a Parker gradient to the bottom, okay. Then wash it and take the chloroplasts bound to the what? Transit peptide and Rubisco small subunit, which is radio-labeled for you, okay. Then you run the gel electrophoresis of that, right? Put some add sample buffer, right? And then run the gel, okay. And then after you run the gel, you dry your gel, right? and then what do? Put Excel film over it. Okay? Radio labeled. Then what do? After X-ray is labeled, it is labeled X-ray. Wherever there is a band, radioactivity, you will see this. Okay? Once you take it out, you will see that one lane you put your only translation product, right? The other is your what? Is your radio where you have done the experiment. So can you go switch to this blackboard? Can you switch blackboard? हो गया यही है ना while running your gel in vitro translation product. So this is your lane 1 is in vitro translation product. Lane 2 is your binding experiment where you did all those things, right? Right? And what you see? Both are in 20 kD. Okay, I told you your mature the transit peptide is 6 kD. You know it. So what? So when 6 kD goes, if it goes imported into the stroma, 6 kD is gone. So how many remaining? 14 kD. So you will not see a 14 kD band here, right? Why? Because you do not do an import study. You did a binding study. Okay. A priori, that protein must be imported into the chloroplast. It must bind to the animal membrane. Okay? And that must happen. We must see that. Okay? Whether you get 20 kD band or not. Okay, that means your precursor protein binding to the chloroplast envelope membrane, to its receptors. Okay? Once you do that, then you do another experiment. Just increase the ATP concentration from 1 millimolar to 1 millimolar. Okay? Assay system. Okay? You take intact chloroplast. Okay? Add your PRSS. PRSS means precursor small subunit. You call PRSS. This is PRSS, precursor of small subunit, PRSS. Okay? All right? Understandable? So you take chloroplast, add your PRSS, okay? Add 1 millimolar ATP. Now there will be import. Okay? Then you do same thing. Okay? Then you do, after import, stop the import. Okay? All right? Simply put it in the ice bucket, it will stop. Okay? At cold 0 degree centigrade, the import is going to stop. Okay? Then you stop the import, put the, whatever you want to do, gel electrophoresis. And if you do that, you will see, again this is PRSS, in vitro translation product. Then it's a small band here. (20 kD). This is 14 kD. Do you see that? You know it? Right. So, what is 20 kD band? This PRSS bound to the envelope membrane, but still not yet imported into the chloroplast. Still bound to the envelope membrane, are being transported to the what? Chloroplast. And this 14 kD band refers to those proteins which is already gone into the chloroplast, whose 6 kD band is chopped up, so become mature protein, 14 kD band. Okay. This comes with autoradiogram. Okay, because measuring the tracing radioactivity only. So other proteins not come here at all. It's a very clean system for you. Okay, to understand whether your translation product, or so-called precursor small subunit, is important in chloroplast or not. Okay. With this minimum assay, first you know that first you clone your gene, do a in vitro transcription to messenger RNA, do a in vitro translation to make a radio in presence 30 methionine or tritiated leucine to get what? The protein, radio-labeled protein. Take this radio-labeled protein, then isolate it intact chloroplast, right? Then add it intact chloroplast to add this translation product to the intact chloroplast, right? Incubate it for 10 minutes, let's say, in presence of ATP, 1 millimolar ATP, then protein import, and you see the 6 kD band, 20 kD band is bound to the envelope membrane, and 14 kD band refers to those proteins who have gone into the chloroplast and whose transpeptide is already cleaved up by the stromal processing protein. Stromal processing protein recognizes where the transit peptide ends and mature protein begins. Okay, that function. Stromal processing protein. Okay, you see SPP is written there, right? SPP, right? Stromal processing protein. Okay, it is nothing but a protease, but simply clips at that point. Okay, that's it. Okay, it's called that we call a processing protein. Okay. So now let's say you want to know whether this protein, the receptor, is a protein or a lipid on the membrane or anything else, right? So what you do, you take the intact chloroplast, isolate intact chloroplast over a Perkel gradient. Okay. Then treat with thermolysin, right? What thermolysin will do? Chew all the proteins present in the outer envelope membrane, but cannot enter the chloroplast. Thermolysin cannot enter the chloroplast. It is a protease which cannot enter the chloroplast, but destroy all the proteins, okay, which are present in the outer envelope membrane. Okay? So take the thermolysin treated chloroplast, okay? All right. Okay, take the thermolysin treated chloroplast, and then what you do? Isolate thermolysin treated chloroplast. Then you do import assay, binding assay or import assay. If a protein is if a chloroplast is treated with thermolysin, okay, then you do a import assay. Okay, then you will see, of course, and the gel you are running. So this is your PRSS, whose band is like this. This is thermolysin treated TL, means thermolysin treated chloroplast. TLCHLP, thermolysin chloroplast, PRSS. But if you see here there is nothing, here there is nothing. No binding, no import. Because all the receptors present in the envelope membrane are destroyed by thermolysis. lysine, right? So what tells you? Prior to this experiment tells you, prior, that not so your receptor is not lipids. Membrane lipids not working as receptor, number one. Okay, because you are being destroyed by protease, right? Not it. So they are not lipids, and the receptors are proteinaceous in nature. They are proteins, right? Not it. Okay. So from this experiment we knew that the proteins, the receptors are proteins. Okay. I am not going details of this. Okay. But touching upon this. Okay. Okay. Receptors are proteins, not the lipids. Okay. Then next question is, there are many experiments went on and on and on. When I'm coming out, I'm talking about the evolutionary discovery of protein import apparatus. Okay. Went on and on and on. Then many experiments were done to see how the transit peptide, so we do not know which protein is binding, right? So what they did, to the they made some anchors, anchored, or flagged. The Rubisco transit peptide was flagged by, and there are some added some binding motifs, extra binding motifs. Okay, and then this radiolabeled protein, and this will bind to the protein wherever it goes, right? I will not go details of it. We will bound to it. Okay, and then they found out to which is cross-linking. So they are simply cross-linking the transit peptide to the protein present in the membrane to find out which protein is going to bind. Okay, let's say it is binding to TOC 159. Okay, covalently bound now. Okay, we made a covalent bridge between the how we made that, all that I am not going to speak about right now. We don't have time for it. Okay, 159 and what 20 kD protein, right? Now the protein that comes out, radiolabeled protein, because Rubisco is radiolabeled. Okay, radiolabeled protein that comes out no more is not 20 kD anymore, but 159 plus 20, 179 kD. Okay, so they found out that the protein is now 179 kD. Okay, that means 179 minus 20 is 159 kD protein it is binding to, right? So they thought that probably this is the receptor to which probably is not completely confirmed yet. Then what they did, okay, they cloned 159 gene and silenced 159, TOC 159 by by mutation. Okay, so TOC 159 got silenced. So this plant does not have TOC 159, right? By mutations. Okay, they got a mutant which doesn't have TOC 159, and they found that this is very yellow, doesn't green, mutant doesn't green, have very little chloroplast development. Okay, and the plastids that isolate from it could not import the chloroplast. They end up bind to the PRSs, precursor small subunit. Okay, many kinds of experiments confirmed. Okay, that TOC 159 is the receptor of the transit peptide. Okay, there are many ways, many things that have, whether the protein is imported like this, or previously we were thinking the protein is going in 3-dimensional form. 14 kD protein, 20 kD protein is entering both the environment in 3-dimensional form and reaching the stroma. We are not thinking before that is being threaded in. This is threaded in, right? We are thinking the protein is being imported in 3-dimensional form to the stroma. Okay, then our knowledge, etc., changed. We did many experiments, proved or disproved our concepts, and later on we found that it is going threaded in rather than going 3-dimensional. I cannot give all the examples here, all the experimental verification of it right now. I have only 1 hour lecture, okay? So, similar for all the components we talk about here. It's not that easy to say, okay? Many experiments went, many people came out, many proteins, different proteins. Finally, certain proteins have stood the experimental test. But I'm telling you, don't be biased by it, okay? We'll tell you in a few moments, there are many issues on this concept. But I give you a consensus concept, okay? Or the proteins associated with the membranes, okay? But tomorrow in a textbook or after a few years, you will see this concept may not be there. Some other membrane proteins will substitute it. Can you go back to the blackboard? The boy is gone. To the yeah, that's the one. Oh, good. Top. Yeah. So all these proteins you see, therefore when somebody has T236, okay, I keep it to be 236 as a complex structure, okay. We'll talk about it if time permits, but I don't think there's time for it. Okay. Now you see. What is this? Transit peptide. I gave simplistic picture. Once a transit peptide, they want the protein synthesized, right? We do this transit peptide. Then it binds to many components. One component is 14-3-3 complex. 14-3-3 protein. It is a class of proteins, okay? Also is a molecular chaperone. Functions as a molecular chaperone. It keeps the protein in transport competent form. As I told you, if this transit peptide, this protein, does not bind to proper what? Molecular chaperones, then it becomes transport incompetent. I mean, it cannot cross the chloroplast membrane. Okay? So we call transport incompetent form. Okay? You see, I have given a topic unsolved questions. You see my title they have given to me. Unsolved questions of Elmer transport membrane. Before I talk unsolved question, I have to tell you what is solved question first. What do we know? Okay. Now this is RBCS, Rubisco small subunit, right? This band HSP70 and 14-3-3 proteins. And this is going to the import apparatus, TOC and TIC. Okay? We put TOC 75, the input barrel, TIC 20 also another input barrel. Okay? Crossing the inner membrane, barrel protein. Okay? This is also a concept. This is, we are putting TOC 33, TOC 159, right, receptors. Here also here is TOC 132. Do you see that? 159 and 132. Okay. They also, the TOC 132 also work as a receptor. We say, we'll see later on, that TOC, the two sets of protein, one is photosynthetic proteins and the non-photosynthetic proteins. That enters the chloroplast, right? The Rubisco small subunit photosynthetic protein, okay? All right. But there are many proteins that goes in, which is a non-photosynthetic function. We have some other functions, but not in, doesn't work in photosynthesis, okay? So photosynthetic proteins goes through input apparatus like this. Non-photosynthetic protein goes in input apparatus like this. Okay. So depending upon which if you only bind to HSP90, okay, then also there is protein import. Here there is only no protein import, but there are many other stories come about it. But we will not go, this is another component could be E1A and Y and all that, don't bother about details, okay. So it's a various cytosine binding proteins and import competence and import apparatus. Okay. First is the various cytosolic binding proteins, whether 1433 protein, HSP70 or HSP90. Okay. On another protein called Y, we don't know its chemical nature very well yet. Okay. And they all bind to it. So the complexity starts here. Okay. So it is saying import competent. What is import competent? So it binds to the protein and goes to chloroplast. It does not bind to the proper import proteins, proper chaperones in the cytoplasm. Then, though all these transit peptides are ubiquitinated, okay, and go to the proteasome complex and getting destroyed. You see that? Okay. So transit peptide alone, okay, does not guarantee protein import. Okay. All right. Many proteins who do not bind to the proper molecular chaperones present in the cytoplasm, they get destroyed. Okay. I will skip these slides for you. Yeah. Now you see. We are saying, what is this? So these are resting, this is import apparatus, resting stage. Right? Okay. Now, transit peptide comes along protein, is not showing you the molecular chaperones binding to it. Transit is there. Now once it binds, what happens? They all are bind with GTP. Top 115 bind with GTP, and top 34 is bound to GDP. Do you see that? For resting phase, top 115 is bound to GTP and top 34. Okay. Both of them, as I told you, are the receptors. Okay, 115 and TUC 34, they bind to GDP. Okay. Now, once it binds, transit peptide binds, you can see it is bound to it. And next step, the GTP hydrolysis, right? GTP is GDP, right? Then one GTP is lost. Then you have to add GTP, so your replenish GTP. Okay? GTP hydrolysis took place. So again you have to put back GTP, give GTP. So okay. So now say this is bound to GTP, and all of them bound to GTP now. Once you get GTP becomes binds. What you in essence is the GTP hydrolysis. Okay? That determines the binding of the protein. GTP must bind, that's with GTP should be hydrolyzed, okay, to do the initial protein binding and import through the intermembrane space. Okay. Ready? Okay. So now it's entering the where? In the tick 20. If you give GTP gamma S, what is GTP gamma S? So, guanine, base, sugar gone in, then sugar, then you have three phosphorus. Last phosphorus is the O bond, you have S bond, sulfur bond. So it is non-hydrogeable. So it cannot hydrolyze to GDP. But GTP gamma S is the compound of GTP which is not hydrolyzed to GDP. Okay? If you get GTP gamma S, there will be no GTP hydrolysis and protein binding import will stop. Okay, I have just finished this. So as I told you, there are complexity of photosynthetic pre-proteins, non-photosynthetic pre-proteins. Okay, so different complexes. Non-photosynthetic proteins, you have TOC 34, TOC 132. For photosynthetic proteins, TOC 159 and TOC 33 and TOC 75. So TOC complex toxic complex remains same. TOC complex discriminates between photosynthetic proteins and non-photosynthetic proteins. Rubisco will pass through this process, with a small subunit, or plastocyanin, or any other protein, okay, that goes through this pathway. Okay. Whereas non-photosynthetic proteins will pass through TIC32, TOC 34, TOC 75 complex, and they both go, and TIC complex remains same for both of them. Okay, they go into the chloroplast. Okay, since your next speaker is ready, the relative abundance of translocon isoforms appears to critical and balancing four distinct classes of pre-proteins required for protein remodeling during the plastic type transitions or response to physiological changes. Yeah. Do it. This is pre-germination, post-germination. You can see protein transport in etioplasts, protein transport into chloroplast. How it is changing. Okay. All right. We stop here. Right. Any questions you have? Any questions?
What we'll do, maybe during lunchtime you can talk to him because the other speaker is here. Now he'll be here during lunchtime. You can talk to him, discuss with him.
Yeah. Okay.
Okay, is it okay, sir?
Yeah, sure.
And I would like to thank again for coming. And I think he's leaving today evening.
Okay.
So big thanks to you, sir, for
Okay.
always helping.
Thank you.
And we have long association with him, almost 40 years.
Right, several years.
Several years. And where we both did PhD with we have commonality.
Yes.
The first student of Prasanna Mohanty and the last but one student.
Yes.
So thank you, sir. Thank you for your excellent talk.
Yes.
And they will interact with you during lunchtime.
Yeah. Okay. All right.